introduction for process development, in conjunction to, what the H3D Institute is doing. I am really grateful for the opportunity, and I'm, absolutely delighted to talk about, some of the work that I did, but also put in the framework in the sense of introducing pharmaceutical process chemistry to the team. So I guess, Susan, you're driving the slide, right? Okay, next, please. So this is a very convoluted map. Some of you guys might have read this, which is called the 4D map for small molecule drug discovery, development and deployment lab, a map for small molecule drugs. So, you have to use a magnifying glass to actually find process chemistry on the map. So, among the many activities, to develop a drug, process chemistry is definitely a small part, but it's a critical and integral part, in getting a successful drug development. Next, please. So there are a number of questions. I mean, two of them are frequently asked. What is API process chemistry? When I was young as a student or postdoc, sometimes I heard it's just scaling things up, making tons of compound, and bucket chemistry is really not that sophisticated. After many years in this business and in this discipline, I have a slightly more refined and nuanced answer now, which is the application of synthetic, physical and analytical solid-state chemistry and engineering knowledge into the development of API manufacturing process. So I think that's probably is a good summary for what process chemistry is. You can look at the 4 pictures over here that will give a good analogy of the role that process chemists play in pharmaceutical development. I know many of you guys are medicinal chemists, very creative people, come up with molecular designs to fit medical needs. It's just like an architect, designing a building for some specific purpose or aesthetic, and put that idea into a blueprint. And the civil engineer has the job to, put the material together, dig the ground, and put the building up. For medicinal chemists, it's pretty much very similar. Chemistry, biology knowledge put into a design of a drug with a molecule. The process chemist has a supporting role to play to transform these design into active pharmaceutical ingredient. API eventually go to finished dosage form that will be able to help patients in need. So process chemist's job is fairly analogous to a civil engineer job in the building site. Next, please. So I tend to go off the tangent when I talk about chemistry. So, I try to put some framework within the talk. So, we will revisit these things, as we go along with the chemistry. So, there are goals, criteria, and skills needed to do process chemistry. The goals are usually fairly obvious. We have to make active ingredient, we have to make API in quantities. The second goal, obviously, is to accumulate knowledge on the molecule, on the drug substance, and the processes by which it's made. So, the knowledge part is important too. But there are criteria to evaluate the effectiveness and the success for process development. My light just switched off. So they are quality, cost, speed of development, robustness of the process, safety, and environmental impact. So, as you can see, some of them are interconnected. And the funny part, of course, is, they don't usually move in the same direction. There are many skills needed. Most of us are trained as organic chemists, but we have to learn a body of knowledge on analytical, physical, solid state, chemical engineering, and very importantly, regulatory requirements as well. Next, please. So I think many of you guys have seen this chart many times. So I just put it up here, but with a focus on process chemistry. So when in preclinical studies, Typically, process chemistry start with preparation of toxicology batch, which kind of lock in purity profile for further development. As we go into first in human or through clinical trial, process chemists, of course, prepare material to support these trials. And the execution and changes in chemistry and processing and equipment really needs to be understood, well documented and justified to regulatory agencies during the clinical phase. If the drug is successful, we'll get to the point where a submission is filed for approval of the drug, but to be marketed. So that is actually what I think is a combination, of process chemistry work. So of course, if that's successful, a new drug will be launched into the market. So I think this is probably one of the ultimate goals for people who are doing process chemistry. Next, please. So, that's kind of the introduction, just give you guys a flavor of, why process chemistry is needed and, what kind of work that a process chemists do. So I just put this up. So today we're going to talk about two molecules to illustrate the point that I just talked about. So, there is one product is a BMS CCL3 antagonist. The other product is a JAK inhibitor, which has the generic name of ruxolitinib from Incyte Corporation. So I just want to go through this real quick because, as a chemist, if you look at molecules, if your roles are different, I mean, you kind of look at them differently. So, if we are in the academic world looking at these two molecules, we may be thinking about, how do we do the best retrosynthesis? What are the stereo control to be put in place and how can we get good papers out of that? Medicinal chemists, I'm sure, you guys will be thinking, how can we speedily synthesize the compound and purify the compound for meaningful assay? But for process chemists, it's a little bit different. So, in the last two boxes, that was my first impression when I look at these molecules. When I look at the CCL3 molecule, I was thinking the synthesis is probably too long. How do we scale it up really fast? But if we do it really fast, it's safe to do it. And can we do a convergent synthesis? And how do we assay the purity because of all the isomers there? For ruxolitinib, when I look at it, the first thing I thought was, how do we source the starting material, which is the bottom part of the molecule, the diazepurine? And, through Suzuki reaction, we have to use palladium. And how do we get it at the end? So, so those were my initial impression. However, many of them turn out to be relevant as we go down the development timeline. However, there are some major hurdles that popped up which are totally unexpected. And these things usually keep me and most process chemists extremely humble. Next, please. So let's go to the chemistry now. So CCL3 antagonist. So, when the team took over the project, we started with preparation of toxicology batch, and then we go to piloting to supply phase 1 and phase 2 clinical supply. The discovery program started at DuPont Pharmaceutical in the late 90s, and then we transitioned into Bristol Myers Squibb and continued development. The drug was intended for asthma. So, I do a very simple retrosynthesis here. You can see the aminothiazole on the left side. In the middle is the chiral amino aldehyde core. That piece is quite hard to make. And the chiral benzylpiperidine, and that's not easy to synthesize either. So next, please. Now, we are getting into the toxicology batch campaign. So the goals are obvious. We have to make enough material for toxicology and other studies, including formulation. And we have to collect a lot of purity data to understand the purity profile of the drug. So the goals are very, very obvious. The criteria for measuring success in this case is definitely the speed of development, the speed of scale up, and the quality of the drug substance being made. So the decision at that point was fairly simple and obvious because we didn't have time, so we had to follow medicinal chemistry for both of the key fragments, and then follow known chemistry to put them together. So the medicinal chemistry is actually very nice chemistry. So, for the amino aldehyde part, we follow the synthesis, from Professor E.J. Corey in the JACS paper in 1996. So, this is a substrate-controlled hydride reduction of a amino amino ester. So that gives a okay level of diastereomers selectivity that will follow by epimerization and removal of the chiral auxiliary. The chemistry worked to give a decent purity of the compound, so we push ahead and did that. For the benzopyridine part, we follow our med chemist synthetic procedure. Our friend George DeLuca used pipridone, do a switch in protecting group. Wittig reaction goes to the olefin in the middle, and then palladium on carbon hydrogenation to give the racemic benzopyridine, which is followed by chemical resolution with our mandelic acid. So, these chemistry work, but they are very tedious. So next slide, please. So, we learn a lot from this toxicology batch campaign, which lasts for a few months. So we find out the benzopyridone is very difficult to source and it's very expensive. So further down the line, this may give us problem in supply. Second point is the racemic pyridine. The resolution of the racemic pyridine is again, tedious and resulting in low yield because you really cannot get any yield higher than 50%. The modest selectivity in the substrate-controlled reduction of the enamino ester actually requires pretty tedious silica gel column chromatography to get the amino aldehyde up to purity. That is a really big concern for us because silica column typically really cannot be readily scaled up to a larger scale. Finally, the API has 3 chiral centers, and of course, there are 8 possible stereoisomers there. So, as I depicted here, with the absolute stereochemistry of the desired product. So we really must control all 3 of the chiral center to high optical purity in order to fulfill regulatory requirement for the API purity profile. For example, if you have 90-10 ratio for one center, is probably too high because even when they multiply out, you may be over the 0.1% impurity standard, judging from the ICH guideline. Next, please. So with those in mind, we switched to a much cheaper starting material, which is valorolactam. So we build the olefin a little bit different, as shown here, to exocyclic olefin coming out from the lactam. So, this substrate is a little bit different from the MedChem substrate. The carbonyl group here actually serves as an anchor, so we think asymmetric synthesis could be possible. Initially we tried palladium on carbon just to see if it reduces, and it does. And later on we tried ruthenium binap. That gives a very modest selectivity, which is not good enough. And the team, myself, we were very frustrated out of that. And we quickly initiated a screen just to see, are we going to find anything or not? Next, please. I remember this was a very good day. So, we screened 32 chiral ligands with 3 different metals for a number of days or maybe for weeks. And then, we find out from one of the 96 well plate, we found this. So the nice simple ligand, BDPP shown here, gave 100% conversion, and it goes with 92% EE. So that was a very good day. So I think the day was good for about 1 day. Next, please. And then I started to worry, will this scale up or not? So, we did a lot of optimization work. So, if you look at the chart, if you look at the ChemDraw here, the bottom half of the chart shows, if we use this iridium catalyst, we are able to get 88% EE just off the hydrogenation. And after that, right, a very simple salt formation with mandelic acid gets the yield to a really good level and also a very high level of, enantiomer selectivity at 99%. So, now we can recall, right, if this piece is at high purity, right, when we combine with the other piece, we have a much better control of the purity profile later on down the line. So, we did a lot of optimization in a 1 liter pressure reactor, which is shown in the purple line. And then because we don't have any equipment in between, so the scale up was actually 400 times to a 400 liter glass line reactor in the green line. So, both of them actually match up pretty good. So, we can conclude the scale up was relatively successful, and we made 21 kilos of the pure, optically pure piperidine. Next, please. So I just want to do one kind of like academic slide because, when you talk about process development too much, it gets a little bit boring. So, Bill Nugent and I look at the results that we have, look at the screen, and we thought it's very surprising to actually get this result. So we move on, with BDPP ligand with iridium, and try to see if this system is generally applicable to other substrate. And the answer is kind of yes. So with different electronic effect on the substituent, different steric effect, ring size, it all worked quite well to give usable selectivity on this type of system. So, we expanded the scope and, we reported it in a paper in JACS some years ago. The fun part really is, when you look at the ligands, over on the left side, right? I highlighted the one that is the best. It turned out to be the simplest, cheapest, and conformationally most flexible ligand out of all of these almost. And the traditional wisdom of having conformational rigidity to the ligand when it combines with the metal center will give high selectivity is kind of not applying here. But in our paper, we quote the Nobel laureate, William Knowles, who is also a process chemist in industry. And he stated seriously, 95% EE is only involved 2 kilocal energy difference. So any kind of prediction before the fact actually is relatively difficult. Next, please. So, we did 1 chiral center now, right? There are 2 more to go. And the aminoaldehyde piece is actually not any easier than the piperidine part. So, as we recall, we used Professor Corey's chemistry for the toxicology campaign. We got some new inspiration from Professor Carsten Baum from Germany. So, he published a paper in '99 in SynLab using anhydride opening with alcohols mediated by natural alkaloid substances and achieve a desymmetrization of these cyclic anhydride. So, we follow his inspiration, we did the chemistry, we got good EE on the ring opening, and then we forced the epimerization to go to the ester-bearing carbon, treat it with a chiral amine and get the salt out conveniently and up the EE to 99%. The next step is actually pretty daring. It's a Curtius rearrangement to take the carboxylic acid all the way down to a protected amine. So you can see, we use an azide reagent, DPPA, to convert the carboxylic acid to an azo acid. And under thermal condition is extrude nitrogen to the azonitrine. The nitrine rearranges to isocyanate. The isocyanate under thermal condition get trapped by benzyl alcohol. And therefore, we have the CBC protected amine. So, all that happens, interestingly, with retention of absolute configuration from the carboxylic acid all the way down to the amine. So after that, it's fairly simple. Low temperature dipole reduction, reduce the ester to the aldehyde, and we isolate that as a bisulfite adduct. Next, please. Now we're putting everything together. So, from the bisulfite salt of the aminoaldehyde. In one pot, we're doing 3 reactions, or in one reactor, we're doing 3 reactions. The reductive amination using step as the reductant, so that we get to the alkylated product in the middle. In situ hydrogenation with palladium on carbon reduced, remove the CBC group, and then the urea formation with the activated carbamate with the thiazole on it give us the API-free base in 80% overall yield in 3 steps in one reactor. So, we all the chemistry went well, but something unexpected happened here, and it caused me and the whole team a big scare. Was that when we crystallized out the free base of the API, a new and different polymorph was observed. Instead of getting nice powder, we got really high density, extremely large colorless crystals. So the purity was good, but the problem is it impacts the solubility of the free base. So, this new crystal has a higher melting point and different X-ray diffraction pattern. So it's definitely a different polymer. So, we have to do some last minute work to modify the salt formation conditions. At the end, we were successful to prepare the fumaric salt, I think in about 11, 12 kilogram scale. That allow us to support the first in human, campaign and further on. Next, please. So, we talked about this chart earlier on. So I keep this on as a report card to myself and the team just to see, how have we done, in terms of the goals, do we meet the criteria, and did we learn anything? So, I think we achieved the goals, and produce enough material at high enough purity. We accumulated a lot of synthetic chemistry knowledge in process testing, purity profile of intermediate and API. We're starting to learn more about the solid form characterization, as I told you about the incident. The criteria to measure ourselves against the quality and the speed of the synthesis, I think we did well on those two. The cost wasn't the top priority at that point. As you could see, I used pretty much all the expensive catalyst and reagent available. Robustness, we haven't really tested, but safety, we did quite a lot of work. So, as a student in organic chemistry, as a student in process chemistry, through this campaign, I learned a lot of organic chemistry, asymmetric reduction, desymmetrization reaction to set up absolute and relative stereochemistry, a lot of analytical method development to enable in-process control, physical chemistry, as you can see, the thermodynamic and the kinetic studies on the Curtius rearrangement is very important to make it safe on scale up, solid state chemistry, engineering, and of course, while we're doing all this, our work is under CGMP, and all the campaign and changes in chemistry and equipment and everything are documented and reported to the agency. The program was not further pursued beyond a phase 2A due to clinical data. So, while it was disappointing, of course, to us and to the company, but it allowed us a little bit more freedom to publish this work. So, those are, shown in the references in the previous slides. So that wraps up the CCR3 program, which I did with DuPont Pharmaceutical Company and Bristol Myers Squibb company. Next, please. So now I moved to a different firm, which is Insight Corporation. So this part of the seminar talks about the process development and the commercial launch of ruxolitinib, which is the generic name of the compound. And this drug is sold as a tablet, as Jakafi, and a cream called Opzelura. So a little bit history on the drug. It was discovered and developed as a Janus kinase inhibitor by Insight Corporation, and we filed the investigational new drug application to the FDA in 2006. That was the time I joined the company. 5 years later, we filed the new drug application, the NDA, to the agency, and was approved for the market. This drug can be used for the treatment of myelofibrosis and polycythemia vera, which is in the tablet formulation, and it can also be used for dermatitis, which is applied as a cream formulation, which is called Opzelura. So the drug has been quite successful. Last year, the annual sale of Jakafi is close to 2.6 billion, and the cream is close to $330 million. So next, please. So in the previous slide, you guys saw a retrosynthetic scheme, and I think it's fairly obvious that the heterocyclic part can be put together through a Suzuki reaction, and then the key part is how do we put the top moiety, on the heterocycle. So you can see there are 3 synthetic routes shown here, and they were all studied in parallel because, as you saw in the previous slide, the development timeline of this molecule is so highly compressed. In 5 years, we have to do everything. So in parallel, we study, all these synthetic routes. The racemic asymmetrical addition followed by chiral separation, that is the original METchem synthesis. It is very direct, and it could get to where we want. But it's probably not the most elegant. The one that coming from the right to the left is organocatalytic asymmetric asymmetrical reaction. So we investigated that as well. And coming from the bottom is asymmetric hydrogenation, followed by functional group transformation. I'm sure after like 20 minutes of this talk now, you guys must be fairly confident that who is proposing this. So, we are studying all these stuff, and I show the references here. The medicinal chemistry is reported in the patent shown there. The asymmetric hydrogenation from our group, from the process group, is reported in this US patent. And the organocatalysis, we reported in Organic Ladder in 2009, and also reported in the US patent as well. Next, please. So now we go into a little bit more, I'll use one slide to describe each of the synthetic route. So it's very abbreviated. Asymmetric hydrogenation. So, if we could make the alpha, beta, and saturated nitrile in the middle, and if metal catalyst in conjunction with chiral ligand can give us conversion and selectivity, that would be very good, because then it will be just one step away, which is the protection to the API. Unfortunately, we screened 22 ligands with 3 different metal precursors, and all of them show very low conversion and literally no selectivity for the alpha, beta, and saturated nitrile substrate. So, with that, it is known in the literature, if the nitrile group is converted to a primary amide, there are a little bit more literature precedent on the reduction of that tri-substituted olefin. So we have some really exciting result on that. So the alpha, beta, and saturated amide, we have 2 results, one with ruthenium, one with rhodium. When ruthenium combined with proprietary biphenyl type ligand, it gave complete conversion at 98% EE. That was spectacular. The rhodium catalyst combined with a ferrocene type, which is similar to a Josephus ligand, also gave complete conversion at 94% EE. So, these are very good initial results. And of course, the problem is the amide has to be reconverted back to the nitrile. That takes some work. Next, please. Organocatalytic asymmetric reaction. So in the early 2000s, I'm sure many of you are aware there was a peaking of organocatalysis. As you guys know, Benjamin Lee, Dave McMillan, they got the Nobel prizes, based on those chemistry. So, we got some really nice inspiration from the work from Professor Carl Jurgensen. So, in this publication in 2007, we took inspiration from him to use our heterocycle, a nitrogen pyrazole heterocycle, adding to alpha beta unsaturated aldehyde, and is mediated by a proline-derived chiral organic catalyst. So, after a lot of screening and optimization work, I'm just going to show you guys the end result. So, we optimize the silyl group of the catalyst. We studied the catalyst loading, the equivalent of the microacceptor, and the acid additive, which is shown in pink here, is also very important. So, we made a study on these factors and come down with the conditions shown in the scheme. So, we got decent yield, 84% yield, and 89% EE on the azomyco addition. So, then we have to take the aldehyde, treat the ammonia, make it imine, oxidize the imine back up with iodine to the cyano compound, and then followed by deprotection. So, all of these reactions have a decent yield and pretty good selectivity. Next, please. So now it comes down to the time for the route selection. Both the hydrogenation and the azomyco addition, I think on their own, if it's an academic exercise, they are pretty good chemistry. That's probably one, some decent publication. But when we select the route, right, there are different factors that we have to consider. So for the hydrogenation, unfortunately, the alpha beta unsaturated nitrile did not perform. So there are some functional group transformation needed later on. So that makes it a little bit unattractive. The second point is actually a little bit more important because of the ligands that are good. They are very costly, and they are also proprietary and have IP intellectual property issues that we have to deal with. So that actually caused us to move away from the asymmetric hydrogenation at the time. The organocatalysis, again, unfortunately for the nitrile doesn't work. It only works on the aldehyde. The aldehyde has to be used in very high equivalent because these reactions intrinsically are very slow. And also, you cannot speed it by heating because by doing that, you lose selectivity. So, high equivalent of the microacceptor has to be used. So, I put the ChemJol here. It is an active aldehyde and it's a microacceptor. So the third point becomes very important. So, if we use such a high excess for this, for the microacceptor, we have to verify the complete purging of this reagent all the way down to the API level, possibly to ppm level. So, that would possibly slow down and draw more scrutiny from regulatory agency. So, we decided also not to pursue this route, even the chemistry is pretty interesting. So, we revert back to medicinal chemistry. So, we hunker down and, optimize the racemic addition, which is followed by a very large scale chiral chromatographic separation. Next, please. So in this slide, I try to summarize almost like 2 years of work in optimizing the original MedChem idea into a manufacturing process. So in green, we show, this is the suitable protecting group to use in the Suzuki coupling. So, you can see the coral diazepyrin coupled with the boronic ester, and the pyrazole is protected as the epoxyethyl. That works well because it's compatible with Suzuki, but it comes off easy. The same group in red protect the nitrogen on the diazepyrin ring. We picked that, among many other options, is that it gives the substrate the highest concentration, which allow us to have a slightly higher throughput for the chiral separation. So, we use a technique which is called the simulated moving bed chiral separation. we work in conjunction with a manufacturer and a chromatographic company to optimize the chiral separation procedure. So from the racemic, we could get to the chirally pure compound with the protecting group on. After deprotection, we form a phosphate salt. We did extensive work on salt selection, and we find out the phosphate salt is the most suitable, and it turned out to have a really good impact on the drug because the permeability and the solubility, the solution rate of the phosphate salt is really high. So that gained us a BCS class 1 classification with the FDA on this drug, so which is very good. The stability of the phosphate salt is really super. I went into the lab myself and grow a crystal for the phosphate salt, and later on, single X-ray crystallography for the first time showed it was in absolute configuration, in the R configuration. So, we clear out all the uncertainty about the absolute stereochemistry, which is very good. And our chemistry team really appreciate the company management because eventually the drug was named ruxolitinib with the R at the beginning to kind of commemorate the determination of the absolute stereochemistry. So as we're developing this process, we have some major chemistry challenges. They are mostly on residual palladium, residual formaldehyde, and residual microacceptor, which is in the API. I mean, these substances are considered genotoxic material. So, we overcome these challenges by appropriate purification method and also very sensitive analytical method to verify their low level after purification. Next, please. I think I'm almost run out of time, but I'll go through this real quick. So this is how we scale this up. So you can see we go from the kilo lab using a pilot column to do the chiral separation to very large scale reactor and also very large scale SMB chromatographic unit to do the chiral separation. You can see in the picture of the SMB unit, there's a gentleman rolling a drum here, so you can get a sense of the scale of the equipment. Next, please. Okay, so this is my long report card, for the whole team and actually for a big chunk of the company. So the ruxolitinib process chemistry summary is here. So in the clinical phase, we are still doing root discovery, which we try to see if hydrogenation, organocatalysis, or racemic route, which one is the best. And of course, at some point, we have to select a route for further development. So when the racemic route was selected, we have to qualify starting material, finalize the route, salt selection, chromatographic conditions, stationary phase. And we also develop a racemization of the unwanted isomer and the recycling of that unwanted isomer to increase the yield and the throughput for the chiral separation. So, all the manufacturing process has control tests for each step. We put a lot of work into the API purification, which I mentioned in the previous slides about the genotoxic impurity that possibly could be there. We need to set and justify API specification in terms of the chemical purity, solid state attribute, particle size distribution, and all these stuff which are important for a drug. And then we move on to the validation campaign. So there are critical process parameter, task, quality attribute that we have to establish. We have to have a final validation of the analytical method, equipment qualification. And then we move on to do a validation campaign, which have 3 consecutive batches run under very highly controlled situation at a good percentage of commercial scale. So all these data will compile, analyze, interpret, and go into the CMC section of the NDA and to be submitted to the FDA. The authority inspected the facility, reviewed our document, and the last bullet point, of course, it was another very good day. The NDA was accepted and approved, and Jakafi became a drug. And actually, the day after the approval, it was launched in the market. Next, please. This is going to be really brief. So we talk a lot about process chemistry today, from the preclinical stage all the way to the commercial stage. So, like as a process chemist, I learned chemistry along the way. And these knowledge are not only applicable to doing proper process development. So this is a slide which shows uh, the knowledge that we gain in the scale up actually could be very helpful to other colleagues within the company too. So, this is a non-process work where we do C14 label with a totally different design of the chemistry. And also, when the label is made and used in human, how these label molecules can help us to track metabolites as shown here. And the knowledge that we have on the synthesis actually expedited the preparation of many of these markers and standards, which really helped our biology colleague, to figure out the metabolism and the PK of the molecule. So this is just one non-chemistry slide, a non-process chemistry slide, which shows, chemistry applicable to the whole spectrum of development. Next, please. So this is the final summary that, summarize what we talk about. We discussed the role of process chemistry, and we introduced the goals, criteria, and skills needed to do this business for two innovative products. And of course, there are a lot of work done on process development, improvement on generic drugs, which are a little bit more cost focused, which we didn't have time to talk about today. The CCl3 compound and ruxolitinib, I want to show you they are really real-life, fast-paced project that I think it illustrates the importance of chemistry thinking and understanding of scale-up regulatory issues. But as much planning that you can do, there's always problem solving along the way because they always arise. And the best way to deal with them is with flexibility and knowledge of chemistry and the regulation. So finally, this is the last bullet. I hope you find a process chemistry as a meaningful, interesting, intellectually stimulating, and really fun endeavor as I do. And along my career, I have learned, as I've shown you, a lot of chemistry from the academic world. We have used Corey's chemistry, Jurgensen's chemistry, William knows, you name it. So, we learn a lot along the way. But most importantly, next slide, please. It is really educational and my privilege to work with this group of people. On the CCl3 hydrogenation, it was me and Bill that kind of invented and developed that chemistry. I finished my postdoc at Scripps with Casey Nicolau, and I was so fortunate to be able to do a second postdoc at DuPont and BMS with William Nugent. He's a good friend and a mentor. I learned so much from him on asymmetric catalysis. The CCl3 process team, the ruxolitinib process team consists of chemists, organic chemists, a chemical engineer, analytical chemist, solid state expert. They are shown here. So, both teams took the project to their respective destination with speed, efficiency, and enthusiasm. I am very fortunate to be part of that. The ruxolitinib DMPK study, I really want to thank the biology team over here in welcoming me into their group to be contributing to their effort from a chemistry standpoint. There are more scientists and engineers working in these three companies that help to make this project successful. And also, the scientists as contract research and manufacturing organizations, they contribute greatly to these campaigns as well. So, with that, I will close, and I really want to thank all of you for attending, and I hope this has been fun for everyone. Thank you.
Thank you, Tai. That was really, really great talk that you've given. I'm going to open it up for questions now. And I guess I can kick off because what quite surprised me, and I wasn't aware before, is that as a process chemist, you've got involved in some solubility optimizations in that one case study. How much of that type of work or formulation do you do as a process chemist?
This is a good question. I think, Susan, you're really leading to something very, very interesting. So, of course, I use about 45 minutes today. So this is an introduction. Today's a little bit heavy on chemistry. So the bridge between process chemistry and formulation, the anchor point is the final crystallization. So, of course, for example, CCl3 actually has more than 10 steps, so all of them are chemistry. But at the end, right, when we get to the salt selection, if it's not a salt, it could be a free form, the polymorph selection, the crystal selection of the final solid. It's a really big topic because controlling the form and consistently producing a consistent form, of course, it's important because it impacts the formulation process. And as we go through the formulation process to the final, the finished dosage form, the solid state attribute of the API would impact the solution rate, PK, and all these aspects directly impacting the patients. There are horror stories within the industry where company could not reproduce their desired solid form and lead to recall, fine by the agency, disruption of supply to the patients. So the anchor point between process chemistry and formulation is really the solid state chemistry. So we, of course, that is a topic for hours and hours of discussion, but I'm really glad that you bring it up. So if we have the chance, we could, of course, have a discussion, with the team on the significance and some of the theoretical background, experimental technique to control, determine solid forms.
Thank you. Are there any questions from anybody else? Otherwise, I've got a few more. One of the things I also wanted to ask about is number of purification steps and how do you do purification on those sorts of scales?
The most commonly used technique for large-scale purification is crystallization because it is efficient, reproducible, and most of the time reliable. There's a large body of knowledge and practices for crystallization as a tool for purification. On the other hand, column chromatography is something that is not efficient to do on scale. And besides that, is something that is a little bit less reproducible upon scale. One can imagine you can do a preparative silica gel column in the lab, which most of us do in graduate school and further on. But that's typically just for grams quantity of material. So physically scale up the column is actually doesn't solve the problem because as the diameter get bigger, right? There were channeling problem and other things that was going on. So, but having said all that, right? I just presented to you that we have a really large scale chiral chromatographic separation. Those techniques are a little bit more involved. The simulated moving bed technique is very intricate and it takes a lot of optimization. And I definitely did not have time to go into that today. But that was a special case. But in general, chromatographic separation could be problematic on scale. So crystallization tends to be the most practical method. And for volatile material distillation is also a very practical purification method.
Fantastic. I can see that there are a lot more topics we can explore. If
there are so many more.
Definitely. I have questions about your environmental assessments as well, and so on. I think there's a lot we could discuss. But if there are no burning questions from anyone else, I would like to thank you so much for taking the time today and joining us quite early in the morning and sharing this. And we hope to be hearing more from you in the future. So thank you.
Thank you so much.
Cheers, Tai.
Bye-bye.
Bye.